Jai Hind everyone. So today's lecture is about what? Today's lecture is about hypersensitivity, and hope so you understood the last lecture what we studied. In the last lecture we studied about the complement system. If you have any doubt, you can ask me. So in this video, we will see the how the complement system will work. Before that, we will continue our last video which we studied already, the cytotoxic T cells. In the last video, we seen that how the T cell will be mature. Okay, T helper cell differentiation we seen. Now we'll see the how the cytotoxic T cell will be mature. So in the if the T helper cell is there, it will release the interleukin, and this interleukin will uh, go and act on the cytotoxic T cells. It will receive by the cytotoxic T cell receptor, and then finally it will transform into CTL. CTL is the killer cell, and it will kill the cells by two manners. Okay, the CTL will kill cytotoxic T lymphocyte will kill the cells. Uh, why it is required the cytotoxic T cell? Because if the pathogen will be escape from where the pathogen will be escape from. Hmm, if the pathogen will escape from the antibodies. So CTL will kill in the two pathogen in two manners. The first is um, first will be according to the MSC molecule. Okay, MSC class 1 molecule or without MSC class molecule. Without MSC class 1 molecule. If it is killing the any cell by class 1 MSC molecule, it means it will it will interact with the CD8 and with the MSC molecule, MSC class 1, and this CD8 will give the information to CD3, and because of this CD3, the these are surface protein only. Okay, these are surface protein on which cell this surface protein will be on the mm, on cytotoxic T cell. Okay, and this will be on the pathogen or in a target cell. This will be on the target cell. CD3 will be acting and it will help in the degranulation. Okay, it will help in degranulation. I will make one small diagram by which you will be able to easily by which you will easily understand how it is working. So MSC1 and without MSC1. So in MSC1, if this is my cell, this is what this is the cytotoxic T cells. Okay, and this is another cell, and this is what target cell. Okay, this is my target cell, and this target cell having the presentation of the MSC class 1, and this is suppose MSC class 1. It will bind with what CD8, and from CD8 it will give the signaling to CD8, then it will give signaling to CD3. Okay, CD3. So once it will give the CD3, CD3 has the cytoplasmic strands, and it will stimulate the degranulation. So the degranulation means what? This have the some granules, and this have the some granules, and this vesicle consists of some enzymes. Small granules will come here, and finally it will degranulate the enzymes. And these enzymes are what? These enzymes will be perforin, per, oh, this perforin will go and act on this membrane. Once it will act on this target cell, the target cell will be form a gap. Okay, not gap. We can see the uh, pores. It will form what? Why eraser is not working? Actually, shit. Uh, so this type of pores will be formed, and because of this type of pores, finally the granzyme will come. Along with this, there will be one some granzyme in there. I'm representing it with this color. I'm showing it with this one more. So this is a granzyme enzyme. Suppose there is some granzyme enzymes also there. These are granzyme enzyme. And once this granzyme enzyme will enters in this cell, it will work on the DNA. Like it will go and degrade this DNA, and finally the DNA will be degraded in the fragments. Okay, the DNA will be degraded in fragment, and target cell will die. This is the one way. Without MSC molecule means what? There will be different receptor which is called the fast ligand, and this fast ligand will be in the target cell. Okay, this just a minute. Let me check. So the target cell will have the fast ligand, and this fast ligand, if you'll see, again I am drawing here the same diagram. I can make it here. Instead of this, instead of the MSC molecule, there will be different receptor molecules that are fast ligand. Okay, what are these? These are the fast ligand. So the fast ligand will act like, suppose this is the cell. Here we send this is cytotoxic T cell. Okay, here also it is a cytotoxic T cell. Okay, so this is the cytotoxic T cells, and this cytotoxic T cells have the ligand. It has the ligand which is called, suppose this is a fast ligand. Okay, fast ligand, and then it has the receptor. Which has the receptor? This target cell. This is our target cell, and this target cell have the fast ligand receptor. Okay, so once it will be interact with the fast ligand receptor, the this target cell will trigger the apoptosis process. Okay, so the like apoptosis will begin, and finally the cell start for the apoptosis, and finally the cell will die. This is the way how this fast ligand will work, and it will go very deep in that how enzymes will be activated, the caspases will be activated, and then how the finally the apoptosis will be induced. That is a different story. Okay, in which we are not going deep. So up to here you understood. Another than this, other than that cytotoxic T cells, the natural killer cells are also there, which will help in the killing. Up to here, we understood the how the cytotoxic T cells will be killed. Now we'll see the natural killer cell killing. Okay, natural NK cell killing. The way of the NK cell killing is uh, almost same like the MSC molecule, almost same like this cytotoxic T cell which will interact with the MSC molecule, but the little things are different. Okay, so the natural killer cells can be activated or inactivated. Suppose this is a natural killer cell. This is a natural killer cell, and this natural killer cell having the inhibitory receptor. What it have? Suppose this is the inhibitory receptor. Inhi inhibitory receptor. If this inhibitory receptor will be there, then the enzymes which it have, the granzyme and the perforin enzymes, it will be not released. Okay, it will not release. It will be here only. Okay, it will not release, and simply it will be here. And if it is interacting with the another the target cell, if it will interact with the target cell, and the target cell have the class one MSC molecule with the inhibitor. This will have in the class one MSC molecule with MSC class one with inhibitor. Okay, with inhibitor. Okay, and there this target cell will also have the another molecule which is called the ligand, or we can say it is a stimulator. If this stimulator will be there now, then it will bind with the another receptor here only. It will bind with another receptor. You can see, and this is the positive. This will be the positive, and like it will stimulatory. Okay, this is inhibitory receptor, and this is stimulatory. So what is happening if both will be there? Like inhibitory is also there, stimulatory will be also there. One negative minus, one negative, one positive will be balanced. Neutral will be there. No release will be there. Okay. So if there will be one it removed, like if the anyhow case the stimulatory receptor will be there, this will be there. But if you will remove the negative, the charge will be what? Positive. If you will remove the negative inhibitory, if you will remove, suppose you remove the inhibitory, then what you will have? The stimulatory you have only. So the net charge will be what? The positive. This positive charge will be allow the releasing of the granzyme. So now this will bind with the ligand, and the granzyme will be released, and the perforin will be released, and this enzyme will finally act on the target cell. Okay, finally act on the target cell, and if it will act on the target cell, and finally then cell will die. Okay, so this is the way how it is working. The important thing is that if the inhibitory and the stimulatory both are equal, then it is okay. But even if the inhibitory will be not there, and any thing will be happen which will be imbalance the negative and positive. If the one negative, one positive is there, so plus minus 0. Okay, and if the plus plus means two receptor will be there, two stimulatory receptor will be there, and one inhibitory will be there, means more positive charge will come. Okay, so it will not work. Okay, so this case, target cell will die. In this case, target cell will survive. Okay, and if the like this, if two negative is also there, like two inhibitory receptor will be there, then also it can be survived. So we have to stimulate this. Okay, if we want to kill the cell, so stimulatory receptor should be work efficiently. Then only we will kill the target cell. So this is about how the uh, how natural killer cell will kill the target cell. Now we'll see the important thing, the complement system. Complement system, I am going to tell you about the at GATV or grade level. So the if you will see the complement system, what it is, the complement system is very simple thing, and this will tell you the about how the innate immunity will develop and the connection between innate and the acquired immunity. But we will see very in very simplest way. Like if I am saying any pathogen is coming inside your body, then there will be no encountering with the antibody in the first reactions. Okay, so the complement system is the only cells which will encounter that cells. Okay, the pathogens. Okay, so the complement system consists of around 30 proteins. Around 30 protein it consists, and this 30 protein is from the C1 to C9, and each have a smaller unit which is represented by A, and larger unit which is represented by B. Got it? So hope so till here you understood. Okay. So each have the 30 protein like C1 to C9. They each have the A and B units. Not you cannot say directly each. Like complement system will have the some proteins will have the smaller and larger unit. 6A, 6B, 7A. I didn't see that for the 7A, but complement proteins will have the larger and smaller units. The smaller unit will be the A and larger unit will be the B. If you will see the smaller unit, there is a one exception for that. The exception is like, what was the exception? That is C3A. Mm, C2A, okay. C2A is larger unit exception, and the C2B is a smaller unit, a smaller subunit. You can, okay. So this is the way how it will be activated. Now, how it will be activated? If we didn't see, we will see it now. I'm going very superficially, like above layer I'm touching of the complement system. I'm not going very deep into this. So the thing is that uh, it starts with the it begins with the IgG or IgM. Okay. So this is the antibody which involve in the classical immunity, which will involve in the classical immunity. So the complement system can be have the it has the three parts. Okay, how many parts itself? I will give you the a brief idea how it will work. Okay, so some proteins, some pathogens came in our bloodstream. Okay, these are the some pathogens. Okay, these are some pathogens. They are looking for the substrate where they can go and where they can sit. Okay, they are pathogens and they are looking for the substrate. And now, if the complement system, if the antibody will be not there, even though if the antibody will be there, the free flowing the complement system which are in our which are in our body fluid, that will be goes and bind here. That will goes and bind here. Finally, it will promote the formation of channel inside the body, inside this pathogen, by which the water will come inside, by which the water will come inside, and finally it will swell and it will die. This is our this is our concept. So the molecule which you are seeing, this is small molecule, na, these are the complement proteins. It cannot directly go and bind this part. Okay, it have to be activates what C5 convertase. If C5 convertase will form, then finally this C5A will come. The C5, the C5A, A and C5B will come. Okay, and finally the C5B will bind, and it will call other protein, and this is the end result, the making of channel by which water will go inside and the cell will swell. So how we can activate the C5 convertase? How we can make the C5 convertase? This is the important thing. Okay. For that we have the three pathways. Okay. For that we have the three pathways. Till here, that all the three pathways will be almost same. So first pathway is the classical pathway. Okay. Classical pathway. In the classical pathway, the IgG and IgM are involved for the activation of the these proteins. In the classical pathway, you will get the C3 convertase for activation of formation of C5 convertase. We want C3 convertase. Okay, you will get the C3 convertase here, which is C4B C2A. Okay, the same C3 convertase will form in the in lactin pathway. Okay, lactin pathway. So this lactin pathway is innate defense. Okay, this is innate defense. Here you will not see any antibody involvement. In this also the C3 convertase will be like this only, C4B C2A. Okay, and finally the alternative pathway. The third is what? Alternative pathway. Alternative pathway. In alternative pathway, what you will see, beta? In alternative pathway, the C3 convertase will be formed. Again, here the C3 convertase will form. In this C3 convertase, it will have the C3 convertase have what units it have actually that. Okay, C3 it will be like C3 B capital B. Okay, so this could be the C3 converters along with the foreign substrate. Okay, along with foreign substrate. So if it will be there, then it will called as a C3 converters. Now this C3 converters role is what? The C3 converters, whatever you seen till now, the role of this C3 converters is important only. It will only activate it will help in the formation of C5 converters. Okay, this will help in the formation of C5 converters. Until unless the C3 converters is not there, the C5 converters cannot be formed. So, so this C3 converters will break this C3, any C3 converters. Okay, Either you found from the lectin pathway or the classical pathway or this alternative pathway. The C3 convertase will work on the C3 protein, C3 molecule, and it will form C3A and C3B. Okay. So the C3B will bind, it will bind and it will form two type of protein. It will form the two type of protein. The it will form help in the formation of two type of protein. Either it will form from the alternative pathway. If it is coming from the alternative pathway, then you will get the C3B, capital B, small b, C3B. So this is our C5 convertase, which you seen in the beginning. Okay. Now, other than alternative pathway, it can help in the lectin or uh, classical. Okay, lectin or classical pathway. It can help in the lectin or classical pathway, and this pathway will give the proteins. The C5 convertase is C4B, 2A, 3B. This is our C5 convertase. Okay, this is our C5 convertase. So finally, we found the C5 convertase. What is the role of the CA? You've seen how where the C3B is involved. Now, C3B is involved in the formation of what? You can say it will not involve in the formation of laddu or something. It is involved in the formation of the C4 convertase. It means it will be very important to get to know what is the role of the C3A now. The C3A will help in the anaphylotoxin. Okay, this is an important behavior, important thing. Anaphylotoxin. So anaphylotoxin will is like a response of our body when the complement system will interact with the pathogen or the foreign particles. Okay. So if the it will interact with the foreign particle, it can give the severe responses. The severe responses will be like the inflammation, increase in the chemotoxicity, like more the invasion of the WBC. Okay, what increase chemotaxis of WBC, increase inflammation, and then it will increase the uh, mm, it will increase smooth muscle contraction. Okay, muscle contraction. So this is all about what anaphylaxis. Okay. Now if you'll see the what is the role of the C5B. Now you the C5 convertase. So this C5 convertase helps in breakdown of C5 into C5A and C5B. Okay, and C5B. So again, this C5A help in the anaphylotoxin. It will help in anaphylotoxin, and C5B will go and bind on the target cell. Okay. It will suppose this is a target cell, so it will uh, start here accumulating. Okay. So this is the C5 B. Now C, it will help in the adding of the other protein like C5, 6. This will be C5, C6. Then C7, C7, little C8, and C8 will be promote adding of what? C9. So finally, you can see this C9 is goes inside the target cell. This is our target cell, and now the more water will come inside. It will make a channel. More water will come inside, and more ions will go outside. Okay, so the ions are will go outside. Okay, so finally the H2O will come, and the cell will swell, and then cell die. So this is the lysis way of the lysis of the cell. Hope so you understood the like complement system. We didn't want to make you confused, so I tried to make you understand in the simplest way, and you can want to devise and you can go back. Important thing is that the classical pathway is the like adaptive pathway, and the alternative pathway and the lectin pathway are the innate pathway. Okay, innate immunity, innate defense. You can write it here. This it will provide innate defense. Okay. So now the point is what? Hypersensitivity. Next topic is what? Hypersensitivity. So hope so till now you understood the videos, and if you have any doubt you can comment me. Okay. Now we will read little fast. Okay. Hypersensitivity is what? The exaggerated response of any for any foreign molecule. What happened to the this? Sometimes it is not working. Okay, I think.